Bismillahirrahmanirrahim. Today we have to talk about the parasites and the role of sialic acid in the infection pathway. It's a kind of continuation of the previous lecture uh, in which we basically discussed about Plasmodium, different types of Plasmodium, and then we talked about some genetic adaptations uh, which actually has resulted uh, because of this Plasmodium infection in human population. For instance, we discussed about different traits like the sickle cell anemia trait, the G6PD deficiency, which these are adaptations, genetic adaptations in human population, which actually are conferring resistance, some kind of resistance to the malarial infection. Today we would talk about this particular topic, the role of sialic acid in the infection pathway, although you can generalize it by including different types of pathogens. Particularly, we are discussing the Plasmodium here as a model for our discussion. uh, what are the sialic acids? So this is very important to understand that sialic acids, all the pathogens take advantage of their presence on cell surface for attachment and infection. Uh, be they viruses or Plasmodium or other parasites or bacteria or different kind of toxins, all these things need sialic acids for attachment and infection because they are present on the surface of all animal cells. And you can say that they are making the outermost boundary of the animal cell. You know, the first thing that other pathogens or other cells would encounter when they come in contact with a particular cell type would be the sialic acid. For instance, if a virus is trying to attack an animal cell, the first thing that the virus has to encounter is the sialic acid. Similar is the case with Plasmodium or any other parasites or bacteria or toxins. If these, pathogens would enter into a particular cell type, that only is possible because of the sialic acids, which are at the outermost periphery or the boundary of a particular cell, shaking hands with things that are coming in contact. For instance, in this image, you can see this is an influenza virus. And the virus, it needs sialic acids for entry. There are lots of research papers describing this phenomenon. You would have seen, you would have gone through it. And this virus, the influenza virus, it has got these heme agglutinins. And heme agglutinins are something that are going to shake hands with these structures. These are the sialic acids, or sials, you can see on the surface of the cell. So these sials and the hemagglutinin have to shake hands with each other before this influenza virus reaches to the receptor on the surface of this cell, and after that it would fuse with the membrane and just inject its genetic material into the cell, which would directly go there into the nucleus. It would take position of the human cell, and then it would make copies of itself, and lots of different types of influenza variants would be generated there. And as the virions, they come out on the other side of the cell, you would find these special enzymes, the neuraminidase, at the surface of the influenza virus, which would cut these newly found variants from the sialic acid, so they would be free to infect other cells. So, these are, you can understand it simply. You should just remember that these are acidic sugars, and these are usually found at the termini of the glycan chains on proteins and lipids that are found on the surface of cells. And the pathogens, they take advantage of their presence on the surface of cells because they attach with these acidic sugars and enter into the cells. So this is a really interesting phenomena because these sialic acids, they uh, are also, you can say, a kind of differentiating animal cells from plant cells and other types of cells, For instance, you can say that plant cells that don't express the sialic acids on their surfaces, or you can say they don't have sialic acids, but animal cells, they do have sialic acids, and there are different types of these sialic acids. If you look at a typical animal cell, you would find out, this sialic acid at the end of these long sugar chains attached to different types of proteins, the integral or peripheral proteins of the membranes, cell membrane. For instance, this is a cell membrane or plasma membrane. This is a membrane glycoprotein that you see here. And these are the sugar chains, these long sugar chains attached with this membrane protein. Or this is another membrane protein, glycosphingolipids. It's a lipoprotein, and you can see these sugars attached with them. These are also attached, the sugar, the sialic acids, they are also attached with free glycans or secreted glycoproteins. They are also attached with secreted proteins. They are attached with peripheral proteins, which are part of the membrane. They are attached with glycoproteins and lipoproteins or integral proteins, which are, integral part of the membrane. So these long chain carbohydrate molecules or sugar molecules are attached with these different types of proteins that are found on the surface of the cell, or which are, even with soluble or free proteins. You know, at the top, you can see these purple structures at the top. These are actually sialic acids, okay? So sialic acid actually is making the outermost boundary of this sugar chain, okay? It occurs at the end of sugar chains, which are connected to these cell surfaces. So this is the end. These purple, you know, shaped structures at the end of these chains. And these are different types of sugars, but these are especially these sialic acids present at the outermost position in these cells. Okay. As I talked about these sugars, these sialic acids, they are differentiating animal cells from plant cells because plant cells do not have these sialic acids. Some people say that they it is so because the sialic acids, they appeared very late during the evolutionary process, this evolutionary process which is going on for billions of years. But some people do not agree with that, as sialic acid are found in some other species as well, which include bacteria, fungi, and some other species. They have sialic acids present in them. The most common forms of sialic acids on mammalian cells are the hydroxylate and acetyl neuromanic acid, which is also abbreviated as NEU5AC, and some people would call it NANA, N-A-N-A. And another common form of sialic acid on mammalian cell is the N-glycolyl neuromanic acids, or NEU5GC. Okay, this is very important to remember, the NEU5AC and NEU5GC. These two are common sialic acids, common forms of sialic acids found in mammalian cells. And these are two different, you can say, conformation of this 9-carbon molecule. It may be found in alpha anomeric form or beta anomeric form. It is found in alpha anomeric form when it is attached to glycans, the glycoproteins. And when it is not attached to glycoproteins, rather it is in some solution form. So 90% of these sialic acids are in the beta anomeric form. So these are two of the, you can say, most common forms found on the surfaces of the mammalian cells. But there is a lot of diversity. If you look at the diversity of these sialic acids that have been discovered so far, there are 50 different types of sialic acids which have been described so far. And these sialic acids, they have a lot of, they have a lot of diversity, but all of them, they have been derived from this particular thing, the neuromanic acid. So they are all derivatives of the neuromanic acid, because, for instance, these two are also derivatives of neuromanic acid. If you look at a neuromanic acid here, it is attached with N-acetyl group, and here it is attached with another group which is N-glycolide groups. Okay, so actually it's the neuromanic acids, or amino group here in the neuromanic acid, which is attaching with different types of groups, which may be acetyl group, glycolide group, acyl group, phosphate group, or methyl group. So when you add a different type of group to the amino acid, it would make a different kind of, you can say, derivative of the neuromanic acid, or a different kind of, or a different type of sialic acid would be found in that case. So you take a one group, attach it with amino acid, the neuromanic acid, it makes one form of sialic acid. Then you take another group and just add it to replace the another group in the, which is attached with the amino acid with that group, it would make another type of sialic acid. So these are different types of sialic acids derivatives. Now we are talking about these two types of sialic acids, the NEU5AC and NEU5GC. So NEU5GC, it is found the highest levels of NEU5, this particular sialic acids is found in mammals. Here, if you look at the phylogenetic tree of the chordates, you would find out mammals here at the top, and NEU5GC is found highly expressed in the mammal, although it is found in other species, other chordates as well. So uh, human beings express NEU5AC, or this form of the neuromanic acid, or sialic acid, but lack Ne5GC because of ALU-mediated exon deletion in the gene encoding cytidine monophosphate Ne5AC hydroxylase. So this is sorry, sorry. So you see that human beings they express Ne5AC or the nana form of the sialic acid, but not Ne5GC because of this deletion which uh, uh, happened in a gene which is called CMAH or cytidine monophosphate Ne5AC hydroxylase. This is an enzyme, and this enzyme CMH converts Ne5AC into Ne5GC. Uh, it means that it's a convertase converting 5AC form into the GC form, one form of sialic acid into another form of sialic acid. So 5GC is part of the infection pathway. It is important to remember, okay? Now, the 5GC, the new 5GC is part of the infection pathway of Plasmodium ricinovi, which infects chimpanzees and gorillas. This form of Plasmodium causes malaria in chimpanzees and gorillas, which is called Plasmodium ricinovi. And this new 5GC is an important part of the infection pathway. Uh, this new 5GC is present on the RBCs, or the red blood cells of chimpanzees and gorillas. It's highly expressed there. And it is required for entry of this Plasmodium species into the RBCs of gorillas and chimpanzees. And that's why this particular species causes malaria in chimps and gorillas. But chimps and gorillas, they have both AC and GC forms of this sialic acid. Uh, because the enzyme that we talked about, cytidine monophosphate Nu5AC hydroxylase, or CMH, is functional in gorillas and chimps. It's not functional in human beings because of that particular deletion which has taken place in this gene in case of humans, but it's functional in chimpanzees and gorillas. It means that Nu5AC is formed in chimps and then it is converted into Nu5GC with the help of CMH. And they would have both of the AC and GC forms of this particular sialic acid. But human beings, they don't have the GC forms because they don't have this particular enzyme. It means that Plasmodium ricino, we cannot infect human because of no GC, because of no new 5GC or this form of the sialic acid in human beings. Because of not having CMMH to convert Nu5AC into Nu5GC. Because Nu5AC is there inside the human beings, but GC is not there. So that's why this particular, Plasmodium species cannot infect human beings, and that's why we say that you know our ancestors in very ancient times they got this kind of resistance against malaria caused by Ricinovi, or a common ancestor between Ricinovi and Falciparum, and by that deletion which happened in the CMA gene. It was a kind of development of resistance against that kind of malaria. So in ancient malaria was caused by this Ricinovi and other types, ancestral types of this Maricinovi. And Fal Falciparum, it's thought that it is a recently evolved species. That's why we say that malaria uh, caused by Falciparum is relatively recent in its origin. And uh, this Plasmodium falciparum has evolved from rich novi strains. One of the rich novi strains probably would have given rise to this falciparum, because when human our ancestors, the hominins, they got resistance to being infected by this rich novi, and then the parasite started changing its strategy because it was allowing 5GC for entry into the RBCs of infected species. And then it relaxed that criteria a little bit by shifting from some during that evolutionary period, which of course would have taken a lot of time, by shifting from 5 its preferences from GC to 5AC. And that also caused this speciation in the Plasmodium species, that from regional falciparum, this particular species emerged or evolved from the rich novi. And this had a changed preference for the sugars found on the surface of RBCs. Uh, so falciparum, Nib5AC is, uh, you know, making part of the infection pathway of falciparum, and Nib5GC is for the older type of Plasmodium species which were causing malaria in our ancestors. So this particular pressure exerted in the human population, which was expanding with time, the human population as compared to other species, uh, it, it has grown over time. So human populations or human settings were sprawling in different parts of the world. So that's why it would have created a lot of pressure on the parasite to evolve in a different way in order to change its preferences so as to capture this opportunity that a great many number of species they are just escaping from you because the parasite would like to find out more species for replication or completion of its life cycle. So human beings, they are too numerous on the surface of the earth right now, and if you compare them with chimpanzees or gorillas. So it has a historical background too. So the parasites definitely would not have missed on that opportunity to, evolve some ways in order to to tap on this particular opportunity to infect human beings. So it changed its way, and in doing so, it evolved into different different forms. Now, if you look at this particular image, it's a uniquely human mutation that causes a uniquely human sialic acid to replace the ancestral form. Uh, this is at the top is Nu5AC. This form of sialic acid is formed inside the human beings, and Nu5GC. This is another form which is found in other mammals. Okay, this one 5AC is found only in human beings. Okay, now if you look at the structure of these two, you would find these different groups at the same places. In case of both of them, look at the carbon dioxide or OR hydroxyl groups. at different positions, you would find all of them, you know, almost at the same positions. And the difference between these two is just an addition of hydroxyl group here. And this addition of hydroxyl group changes the form of the sialic acid from Nu5AC to Nu5GC, from, you know, acetyl to glycolyl. The CMAH enzyme is actually doing this particular job. It is adding another hydroxyl group here just to make or convert Nu5AC into Nu5GC. So who is doing it? This particular enzyme, the CMAH enzyme, is doing it. It's doing so in chimpanzees and gorillas and other mammals. But as we talked about in human beings, you know, this particular thing cannot happen. T5AC, this addition of extra OH group to this would not happen there because the human being CMA gene has been inactivated, and that happened 3.2 million years ago. So it's not a recent story. As I told you, that CMA gene, it was inactivated, you know, long ago in our ancestors, the hominids, you know, 3.2 million years ago. You know, you can say that our ancestors, they evolved this particular trait, an activation of the same CMA gene. This particular trait was selected among our ancestors because it gave us protection against uh, malaria caused by rich novi, Plasmodium rich novi, which was prevalent in the ancient times. Or the different strains of rich novi are their ancestors. They were causing malaria, this deadly disease in the human population. So that trait was selected, it prevailed in the human population, and we got rid of malaria for many years, for thousands and thousands of years. We did not get any malaria. So if you look at this particular, you know, graph here, these are uh, million years before present, 1, 2, 3, 4, 5, 6, 7, 8, okay? And this is the tree, uh, you know, uh, showing a hominin events or parasitic or plasmodium event and anopheles mosquito event that millions of years of evolution, this evolutionary story is being depicted in this particular picture. So where did we evolve this trait? The CMA gene was inactivated. It was right here during the hominin evolution. The loss of Nu5GC target, it happened here. And initially, these malarial parasites, they had established a very long association with our ancestors. They were causing this problem, and this severe uh, malignant form of malaria appeared uh, in, you know, during these four, uh, between 3.5 to 4 million years ago. And here, at this particular found, around 3 million years ago, or 2.5 to 3 million years ago, this loss of Nu5GC target actually was, it was noticed that, or it has been estimated that this particular change in the CMA gene would have taken place during this particular era. So it broken the malignant sign of malignant, cycle of malaria altogether here, because malaria was evolving along these different species. It was, the Plasmodium was not only infecting gorillas and chimpanzees, and were also infecting other species. But the malignant cycle, or more, severe cycle of malaria started here. Our ancestors definitely suffered during this particular period. But after suffering came this uh, change and this inactivation of the gene which is required for this particular purpose. And then we had a long period before, you can say, for millions of years, our ancestors, the hominins particularly, they did not get any malarial infection during this particular period. And when this parasite felt the pressure that hominins on one hand are increasing on the surface of the earth, these species are decreasing on the surface of the earth, so it needed some kind of shift in its strategy to evolve along different lines in order to change its preference from Nu5GC to Nu5AC. Uh, so exploitation of the Nu5AC as a sole target was attempted during this evolutionary period, which gave rise to a different form of the Plasmodium species. This is speciation which is taking place due to this evolutionary pressure, and this speciation resulted in the formation of Nu5, in the formation of Plasmodium falciparum, which is causing more severe form of malaria nowadays. And it's a kind of egoistic, revenge that we avoided attack by the Plasmodium species for many years. So you can also go through this paper, human-specific evolution of sialic acid targets, explaining the malignant malaria mystery. This is very important for you to go through and study about these human-specific evolution of sialic acids in different settings. And there is another thing that you should remember about the sialic acids, that these sialic acids, they are not only doing this particular function, that they are involved in attachment of different pathogens with the cells. The cell-to-cell attachment, if the cells are attaching to each other, again sialic acid, as they are present on the outermost surfaces, so it is important for all those cells to attach with each other, using these sialic acids. And cells in different tissues or different organs, they have sialic acids of different kinds. And those sialic acids are keeping cells of the kind together. It's like birds of feather flock together. So cells with similar sialic acids, they would just stick to each other in different tissues. For instance, the liver cells or spleen cells or heart cells, or etc. So these cells stick to each other because of this particular capability, or this particular quality. They have similar sialic acids on their surfaces. So there is bad news for the red meat consumers here. As we are talking about sialic acids, so we should also discuss this particular thing, that the red meat consumers, they can contract these, they can suffer from autoimmune disorders or autoimmune diseases. Why? For that you have to understand this thing, that human gets Nu5GC from diet. As we discussed earlier, that human do not express Nu5 GC. NIV5 rather, NIV5AC is expressed in humans. So where would the humans get it from? The humans get it from the diet. And the main source is the red meat or the mammal's meat, which is rich in NIV5GC. It's not present in plants, so we're not getting it from plants, and that is for sure. And what is the problem? The problem is that because of higher similarity between 5AC and 5GC, as I show you the diagram, there is a difference of only one hydroxyl group, okay, between the two. So it means they are highly similar to each other. So the difference between the two is very minute. It means that the GC form, SNE5GC, would be absorbed and become part of the human cells. When you consume meat, for instance, or mammalian meat or the red meat, which is rich in the 5GC, GC would be absorbed and it would become part of the human cell. And when it becomes part of the human cell, the body's immune system, still considers Nu5GC as a foreign body. It doesn't belong to the body. It was just, uh, you know, um, mistakenly absorbed and utilized to become part of the human cells, is making their outer boundary, but it's not actual part of the human cell. And that's why it would be considered as a foreign body by your immune system. And antibody response would be generated, humoral response would be generated against this particular JC form. And this kind of antibody is called a self-antibody. This condition is autoimmunity. So people who are consuming more red meat are more prone to develop these autoimmune conditions, which include cardiac conditions, heart problems, kidney problems, different types of cancers, and transplant rejections. Particular people who have received any kind of transplant, uh, kidney or some other transplant or skin, something, they should avoid red meat because that can actually increase the chances of rejection of that transplant. You can also go through this particular paper, human uptake and incorporation of an immunogenic non-human dietary sialic acid, from where you can get very important information about the sialic acid. And this is a diagram showing all this that we discussed here. These are the guys on the right side. You can see these animal species. They are rich in the GC form, and when you eat uh, their meat, particularly the meat, okay, it means that you are eating the GC form, NIV5GC, okay, and this NIV5GC would be absorbed in your body. It would be incorporated inside the cells of your body. What is incorporated? What would happen? Antibody response against these would be generated. Anti-GC antibodies. Anti-GC antibodies would be generated in your body, and you know this would also invite the T and B cells, or they would also overreact because these cells have to undergo this scrutiny, which molecule is self and which is non-self. So the T and B cells would also be activated. Humoral response would be activated, and the GC form would be, targeted, for elimination. And that elimination actually requires different lines of your defense system, including cellular-mediated and humoral responses. So, antibodies will also be generated against your own cells, because these are mounted, the GC forms are mounted on your cells. So antibodies and the cellular mediated immunity would target your own body cell. It would be a chronic inflammatory situation. That inflammation would be going on and on, uh, you know, uh, which would lead to uh, the risk of cancers or heart attacks in different individuals. Similarly, you are not just taking it as food. You can say that we are using different products coming out of the animals industry. For instance, serum substitutes, the serum substitutes of animal origin, or the animal cell lines that we are using. We are producing different types of vaccines and different products in the animal cell lines. Similarly, stabilizers, all these things, the therapeutic things, vaccines or other therapeutics, in the shape of plasma, etc. Uh, these antibodies and cytokines, enzymes, hormones, these are produced using cells or systems based on animals. And there's no way of purifying these things to, a level where you would be confident enough to say that they are 100% free of the animal debris. So all these things, on the other hand, apart from these impurities, there are GC and the 5GC of the animal origin incorporated in these things. And it also goes into your body and generates the same kind of response. So people who are taking these animal meat, it increased risk of heart attacks, cancers, and other autoimmune conditions, including renal failures, alopecia, etc. So you should be very careful. I think it's enough for today. You should remember that uh, what is the importance of 5GC with respect to malarial infection and how speciation uh, took place as a result of mutations in a particular gene in human beings. So next time, see you. Thank you very much for your time, and I hope you can ask me questions later on. Um, you can also, I think, join a meeting where we can have a meeting where you can ask questions about the slides and we can discuss different things. Thank you very much.